Hello my access to HE little chimpanzees. Welcome to the next edition of these screencasts. Today we're going to be talking infectious disease and specifically the specific immune response. Let's start with a recap as we normally do. The human immune system is built on two distinct sections: the non-specific or innate, and the specific or adaptive. Now we've covered this in great detail already, but this, if you imagine, is our castle, our immune system, or part of our immune system that is ready to go at the drop of a hat. If we come into contact with any pathogens, our non-specific immune system is there to deal with it. So we said that there's barriers to infection, like our skin, our mucous membranes, our stomach acid. Are like the walls and moat of a castle. They stop things from getting into the castle. And then if these attackers get over the wall or through the gate into the castle, then the alarm bells ring. The alarm bells in our body is the inflammatory response, which brings the troops to the battle. When there's actually fighting, don't know what that means, but if there's fighting in the castle, that's when our non-specific immune white blood cells perform phagocytosis, and that's the actual battle itself, the fighting of the battle. They don't know what it is, they've just waken up, they've just been flung outside their barracks, and they're going into battle. If you don't understand those three things, go back to the videos, make sure you do, because it does link. They don't operate in silos, so it's not just a case of that the non-specific immune system works and then hands it over to the specific immune system. There's consistent interaction between the two systems, obviously because we want to maximize the opportunity of killing pathogens. So today what we're going to explain is the specific or adaptive system, and this is where the trained killers come into play. These guys who have just been awoken out of the barracks, the general sentries, they're a bit dim, they're powerful, strong, and they'll kill anything that they need to. They'll throw themselves into the battle, but they're not very clever. They're not trained, whereas these cells, the cells of these systems, are highly trained killers. And what we need to do is to understand how they work and how they're working in your body. So let's go for it. This sort of video accompanies a handout that I publish, which is the drawing canvas. So you can fill it out as we go, or you can create your own. It doesn't matter. But I find that this sort of table gives a nice sort of reference point to when we're learning it. So you can see at the top there, this is the summary of a specific immune response and the main players. We have got the humoral response here down the left and the cell-mediated response. What I need to do first of all is set the players in motion. I need to tell you what the pieces of the board are, almost. And then in this review section, I'm going to put it all together to give you the bigger picture. Okay, so the humoral response is generally related to the humour, and the humour is not the ha 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 funny, it's the fluid around the cells. So humour is fluid. And the cell mediated tends to be cell-to-cell communication. There are two major cell types that are involved in these responses. The cell type that is responsible in the humoral response is a B cell. Now, to give it its full name is a lymphocyte, so it's a B lymphocyte, but I'm just going to call it a B cell. You do need to know that it's a lymphocyte. And a lymphocyte is a white blood cell. It's a leukocyte. It's a type of leukocyte, and don't get me started. It's very complicated. But the cell-mediated response is concerned with T cells or T lymphocytes. I'll put the S on because there's more than one. Okay, so B cells and T cells, if you hear those, then you're going to be explaining or describing or concerned with the specific immune response. Now, they're both born in the bone marrow, and we know all about that from our skeleton and muscles unit, if you've done that So they're born in the bone marrow, and when they're born, they have to pick a receptor. Basically, they pick a shape of receptor. And again, if you've done the macromolecules unit, you'll know about that receptors are proteins, and proteins have a specific shape, and that specific shape is determined by the genes or the genetic information. So B cells pick their receptor, and their receptor is a Y-shaped protein as such. It's not a great diagram, I have to admit. I'll do another one over here. A Y-shaped protein, that is meant to be a Y. And these Y-shaped proteins are antibodies. And these antibodies, which I'm drawing here on the end, have receptors on them. They're specific shapes, and they're made of protein, just like we've learnt about. So B cells have antibodies in their cell membranes as receptors, whereas T cells have the old classic T cell receptor sort of thingy-majiggy going on. In their cell membrane they have T cell receptors, and they're specific shapes. They're made of protein. They have they're coded for in the DNA. They get transcribed and translated, just like we said in protein synthesis. Now, where do they go to school? They have to go to school to pick these receptors, right? To pick these receptors, and they do that. The B cells do that in the bone marrow. B for bone. So B cells are educated in the bone marrow, and the T cells are educated in the thymus, which is a gland around the heart somewhere. Okay, so that's where they get sort of schooled, and they have to go through classes. So they pick a receptor, which we've just said. Now that, you know, if we're talking about this receptor here on the T cell, there's, you know, X number of shapes that protein can be. We oversimplify it in sort of science when we're learning it into these nice and easy shapes, but they can be, you know, if you have to think three-dimensionally, they can be really extremely different shapes. And so these receptors are very specific. But when they come out, when they pick their receptor, we describe them as naive. They're naive, which means they've not really been in battle yet. They've not fought a pathogen. You know, they've come out, they've been schooled, they've been given their receptor, but they're still naive until they actually interact with the pathogen, until they have some killing until they've earned their stripes almost. They're still called naive, and they go through something called negative selection. Negative selection. And negative selection, these are both cells, by the way, B and T cells. They go through negative selection, and they get tested in these classes. They get tested as to whether they will bind to their own cells. So if you can imagine, if these white blood cells bind to your own cells, they're going to cause a problem. They're going to start destroying your own cells. So we can't let them go out and round the body start eating up your own cells, because that's just going to be no good, is it? And we call that self-tolerant. Are these receptors self-tolerant? Do they tolerate your own cells of your own body, or are they going to cause problems? And 95%, so I'm led to believe, is not self-tolerant, so they get destroyed. Only 5% of people pass, or these cells pass the class and are allowed to leave. So it's a 5% pass rate. 95% of the white blood cells get broken down and recycled. So that's negative selection, and that's testing whether they're self-tolerant. Will they bind to themselves or not? We don't want them to bind to themselves. Now, T cells also go through another class. They go through positive selection. I'm going to do that because it's just easier. Positive selection. And T cells have to bind to our good old Twitter, Facebook, social media of the cell, MHC. That could be MHC1 or it can be MHC2. But if these T cells are going to be any good, they have to be able to bind to MHC. If they don't bind to MHC, then they are useless and they get destroyed. Okay, so that's positive selection. So not only do T cells have to be able to not bind with themselves, they do have to bind with MHC. Otherwise, they're completely useless. So if you missed any of that, rewind it, recap it, because now I'm going to go to the big picture. Now that we know really about our main players, I can then start talking about how this specific immune system works. So in the bottom right hand corner here, I'm going to draw a body cell, and this can be any sort of cell. Let's say it's a epithelial cell of the trachea. Right. Now, any cell with a nucleus is able to go through phagocytosis. Right. Any cell. Right. And if you remember that any old body cell, it presents its information on MHC1. MHC1. We know it's 1 because it's got one sort of one connection to the cell membrane. That's just how we remember it. So that's how a normal body cell connects or informs the local environment in the body about what's going on. Okay, and it's chomped it up, it's posted it on the cell membrane, and it's gone, look, I've just got this purple virus, purple bacteria, whatever it is, what should we do about it? Now, when we talked about phagocytosis, phagocytes, and I'll draw one here and label it a P, we said that they were non-specific white blood cells. And that's right, they're non-specific. They do eat pathogens, and that's their specialised job. That's their specialised job. And differently to the body cells, they are able to present pathogens that they eat on MHC2. That was what we said in phagocytosis, and if you're not fully aware of that, then go back to it. But that's the main difference between a non-specific white blood cell doing phagocytosis and a regular body cell doing phagocytosis. And what's this got to do with this specific immune system, I hear you say? Because I've not talked about B or T cells. Well, let's have a look at our T cell. Here's a T cell, and it's naive. It's just come out of school. It's picked its receptor protein or T cell receptor. Try and draw this as best as I can. I'm doing it with a mouse. And I'm not going to shade everyone's in, but there we go. There's its T cell receptor. Now, you'll notice that this naive T cell's receptor matches the MHC2 complex on the phagocyte. Now let's just take a second there, because what we've just said is that T cells have to bind with MHC. They have to bind with MHC. So this is where it comes in. The naive T cell binds with the MHC2 on a phagocyte. Remember, this is non-specific. This is part of the non-specific system. I'm going to run out of space, but never mind, you get the idea. So when that happens, the magic happens. The naive T cell no longer becomes naive, it becomes an activated T cell. An activated. You've just turned on the Terminator. It knows what its target is, and it's now being authorized because it's got a specific shaped receptor. Yes, that is the same shape as the one above, because now it can do its thing. And when it does its thing, it differentiates between 3 different types of T cell, through mitosis and genetic turning on and whatnot. So this Terminator then can turn into a killer T cell. I love that name. a killer T cell, often called or sometimes called a cytotoxic cell. You might see that in your reading. These all have the same receptor proteins on because they're just going through kind of mitosis. They've been passed on, they've been authorized to use this receptor protein now because it's been activated. And the killer T cells roam around the area, and if they find an MHC1 complex with the matching antigen, with the matching bit of pathogen, then I'm afraid it's time up for this body cell because it commits suicide. And that's just amazing. That's a fantastic little feature, because if this cell's presenting this virus and this naive T cell's been activated because it knows there's this virus in the area, then this body cell, on every other body cell that presents this antigen on its cell membrane, on its MHC1, will just self-destruct, and it will try and not allow the virus to replicate and replicate and replicate. Now that is unbelievable. I think it's awesome, and what a fantastic thing for our human body to be able to do. The second cell that my activated T cell can turn into is a memory cell. A memory cell. And a memory cell is one that lasts in your body for a very long time. You know, it remembers the activation. It remembers that we've had this thing before, and it stays around in your body for a long time. Okay. And I'll talk a little bit about that on the next slide. So a memory cell is one that has been activated. So it's not a naive T cell. It's not one that's been activated in the bone marrow. It's one that's been activated already and is floating around your body, waiting to find the right pathogen and the right antigen. Because if this thing gets activated again further down the line, it's going to instantly trigger this killer T cell, or this other cell that I'm going to talk about now, which is a helper T cell. A helper. In fact, let me just delete that. I think I need it a bit higher. Let's draw it up here. A helper T cell. Okay, and that's the third and final cell type that it could differentiate into if it was activated. Okay, so the T cell binds with the phagocyte, a dendritic cell or the like, and it gets activated, and it activates into one of three different cells: a killer T cell, a memory T cell, or a helper T cell. Well, what does a helper T cell do, I hear you ask? Well, this is when we encounter the naive B cell. The naive B cell. Goodness me, I'm writing terrible, but you get the idea. Now, the naive B cell, remember, has an antibody ingrained all over its membrane, and that has a specific receptor on the end of it, and that receptor, you'll not be surprised to know, is the same or very similar to the pathogen on what would have been on this this phagocyte here. So in order for this phagocyte to phagocytose, it must have been able to bring it in through phagocytosis. So what happens is that this naive B cell binds with the pathogen. So let's draw that in red. Let's draw this as a virus, for example. There's a virus, and there's its spike proteins on it. Blah, 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 blah. And what happens is that this naive B cell then presents it on MHC2. So let's do the same colour. So you might have missed that in translation a little bit there. So here's the pathogen. here's the antibodies poking out of the cell membrane. And what's happened there is that this naive B cell has taken in the virus, phagocytosed it, and presented it on its MHC2 complex. And this is where the helper T cell comes in, because the helper T cell, remember, has to be able to read MHC2. And it does. It reads MHC2 on the naive B cell. And that's how the naive B cell becomes, I should draw that in green, activated when that happens. So activated B cell. So that's crazy, because all of this here on the right hand side has to happen in order for the B cells to be activated. If this stuff doesn't happen on the right hand side, then that B cell does not get activated. So in order to get a full-on immune response, it goes through a lot of sort of checks and balances to make sure that this happens. And hopefully you can see that that T cell receptor binds with MHC2 on the naive B cell, which then becomes activated. So what happens when this B cell becomes activated? Well, firstly, this B cell can become a memory cell, just like the T cell. That does say memory. That will stay in your body for a long period of time, waiting to interact with the pathogen once again, the old enemy. The other specialization that can happen when a B cell is activated, it can turn into a plasma cell. And a plasma cell goes into overdrive. Basically, it goes through protein synthesis like no other, and it starts producing these antibodies. And I'm talking like thousands a second. I've forgotten the quote about how many, but it's ridiculous. It floods your system with antibodies. And not just any antibodies, the antibodies that are going to attach to the virus that they've been trained to attach to. Okay? So those antibodies, as we know, as I commented on in the phagocytosis, they can start clumping, they can start attaching to the viruses, and they can chain viruses together. They can tag them. Basically, they tag them for destruction. So what can happen is now then the heavy boys, the dim boys who haven't been to school, but they do have receptors for these, they do recognize these antibodies, they can start just chomping up all of these pathogens around, because they've just been lit up by these antibodies. These antibodies are sticking to the viruses and the bacteria, excuse me. And they just allow these phagocytes to start doing their work. And there's the link there when I said on the first slide that there's definite links between the non-specific system and the system. You can see it all down the right hand side, you can see it on the bottom there. It all works together to protect our bodies from harm, which I think is absolutely fantastic. So not only do we store antibodies, we make memory cells for the future. We also have killer T cells which go around and just make cells self-destruct if they're showing any signs of that virus or bacteria around. And also we have these phagocytes which just, once it's been targeted, once it's been highlighted, can then go and work their magic. And that is the specific immune response. And as you can imagine, the specific immune response, like we said in the early videos, takes a bit of time to get into motion. The non-specific system's there, it's immediate, it's fast, but it's less powerful. This takes days, sometimes weeks, but once it's in operation, then it's like a nuclear reaction and it completely destroys anything that's not meant to be there. Okay, let's take it a little bit further in that we are going to look at a typical exam question. On the left hand side here, you've got a graph showing something, an antigen, let's say, or let's recap, An antigen is something that triggers a specific immune response, right? So this could be, it could be an immunization shot, for example. And you can see that the latent period, the period in which it takes to kick off an immune reaction, is 2 days to hit the peak of its response. Now, when we get the second injection, we can see it starts here, and it already reached its peak in under a day. So this takes 2 days, but this is under a day. So the second response is under a day. Okay. So the primary response, the primary immune response is happens when the immune system encounters an antigen for the first time. That's the primary immune response. When the naive T cells and the naive B cells have to get activated. And they have to go all through those checks and balances in order for it to start to work. The secondary immune response is the second time and any other subsequent time when the specific immune system encounters those pathogens. But this time there's no real activation because the memory cells are already there. They've already gone through the checks and balances. There's no need to go through all of that process. They have the memory cells. They have the receptors. They know that they're self-tolerant and they are self-tolerant and immunocompetent. Immunocompetent. That's what it means by they combine to MHC 1 and 2. So they don't need to go through all the checks and balances, and which is the reason why we get, first of all, a quicker response. Quicker response in this secondary immune response, but also look at the amount of antibodies produced. You can see it's massively improved this second time around. So you can see that those plasma cells are really able to differentiate fast, and they're able to differentiate and also be able to create more antibodies, which again helps the non-specific immune system. Okay, I've put a link up there which kind of is a nice summary, which kind of explains that and summarizes it. But basically you've got that the primary response is the first time that the immune system encounters a antigen. The secondary immune response is the second time and every other time that the immune system encounters the antigen. The secondary immune response not only provides a quicker response, but it also provides a bigger response in the number of antibodies that are being produced, and therefore we are able to deal with pathogens and infection a lot quicker the second time that we encounter them. Hopefully that makes sense. So the next question then would be, explain how a vaccination works. How a vaccination works. Well, a vaccination basically readies the body to trigger the adaptive immune system, or the specific system, whichever one you want to call. Immune system. Goodness me, I get too carried away, too excited. So a vaccination readies the immune system, the specific immune system. It preps it before it even encounters it. So it kind of reduces that primary. It's also it's like an artificial primary immune response, so that we can skip the primary immune response. We can get to the secondary one and get those antibodies and that reaction out there a lot quicker. You're still susceptible to having a virus a second time, but the fact is that your immune system responds that much quicker, and therefore you're less able to suffer from serious health consequences. So there are different kinds of vaccination. We have live attenuated vaccines, which are weaker versions of the pathogen. So they're sort of adapted somehow in that they're not as infectious. We also have inactive vaccines. Inactive vaccines, where basically they're killed, they're destroyed versions of the pathogen, which can still trigger a specific immune response, but they are not able to give us the disease. They're not, we don't get we don't even get, we don't get any problems with in the human body, because they're either killed or they're weaker and they're not going to give us that much of a response. Okay. We also have a subunit vaccine. That might be just like giving them the spike protein or a load of spike proteins of the virus, which again could be the antigen to triggering the immune system. So a subunit just means part of the pathogen. It's the part that the immune system identifies. That's all we need. We only need the part that binds to that receptor, and Bob's your uncle. We can start training our immune system to deal with it. And I'm led to believe scientists are working on DNA vaccines, and DNA vaccines are cutting edge. Basically, they are basically a bit of genetic material which are injected into us, which then have the corresponding information to correspond to the shapes of the proteins on the virus or bacteria. So it allows us to create the right receptor proteins, essentially, to be able to identify these pathogens a lot more effectively. But in summary, vaccinations ready our specific immune system, and they're almost like an artificial primary response. So that comes on to the different types of immunity that we can get, and you're asked to do this in your booklets or your assignment. We've got to fill the table in. We can, so we can have naturally acquired active, naturally acquired passive, artificially acquired active, and artificially acquired passive. Now, an artificially acquired vaccine might be a vaccine, might be what I've just talked about. It's artificial and it's active in the body, causes an active response. Whereas we can get immunity through childbirth. So a newborn baby could get antibodies, etc., through the placenta. So it's naturally occurring, and it's also a passive form of immunity. Now finally, what we could say is that artificially acquired passive, it's artificial and it's a passive process. We could inject some sort of blood products that have sort of immunoglobulins in it. It's artificially acquired, but it's also a passive. It's a passive sort of immunity rather than active. I have included a link there, which is quite nice. It summarises it, but go ahead and by all means use your own a little bit of research to see the difference between what active and passive means, and that will not take you too much. I hope you get the gist. I hope I've done a good job there, because it's not an easy topic. It's quite a big topic. In 30 minutes you can rewind it. You can use your canvas if you've got it, or you can create your notes so that you can relook at them and go through them. But don't worry, it does take a bit of time to sink in. It is complicated, and also if you do have a grasp of phagocytosis and inflammatory response, you do get the big picture of how the immune system works. All right, that's it. Chris out. Hope that was okay.